Howdy, my name is Jasmine Gonzalez, and Natalie and I will be presenting over cell-mediated immunity. I will be going over Chapter 5, Functions of Helper T cells versus Cytotoxic T cells. So we have helper T cells, which recognize antigens that are presented by MHC class II molecules on antigen-presenting cells. These cells activate and regulate immune responses through the secretion of cytokines. Some examples will include interleukin-2, interferon gamma, and interleukin-4. These cells also aid in B cells in the production of antibodies and cytotoxic T cell activation. These cells can be broken down into further subsets like Th1, Th2, Th17, TFH, and Treg. Cytotoxic T cells recognize antigens presented by MHC class I molecules presented on all nucleated cells. These cells are responsible for directly killing virus-infected tumor cells and bacteria. These cells also secrete interferon gamma for replication and enhancing macrophage function. Lastly, these cells are able to induce apoptosis in target cells through two main pathways, which are known as perforin granzyme pathway and Fas-Facil pathway. Next, we have the steps in the activation of T lymphocytes. This is where T cell activation is a complex process, and it involves 4 different steps in order to ensure that T cells are properly activated. First step is antigen recognition. Here, T cells use their receptors in order to recognize antigens presented on MHC molecules. So we have our CD4 plus T cells, which recognize the antigens of MHC class II molecules, and we have CD8 plus T cells, which recognize the antigens on MHC class I molecules. Next step we have is co-stimulation. In order to fully activate T cells, a secondary signal from the co-stimulatory molecules on the antigen-presenting cells is required. CD28 binds to our B7 proteins in order to amplify TCR signals. CD3 transduces these signals into the cells. Next step we have is cytokine secretion and response. After activation, T cells secrete cytokines, which can stimulate further T cell activation and control inflammation. Cytokines are crucial for infection elimination by cell death induction. Different cytokines each have specific differentiation pathways. Some examples are IL-12 to Th1, IL-4 to Th2, and etc. Step 4 is proliferation and differentiation. So T cells proliferate and differentiate into effector T cells or memory T cells. We have the effector CD4, effector CD8, and of course our memory T cells, which provide long-term immunity. Signals needed and cellular receptors and ligands that are used to activate T lymphocytes. So we have 3 signals, signal 1 being antigen recognition, signal 2 being co-stimulation, and signal 3 being cytokine signaling. After recognition of the ligand by TCR chains, CD3 is involved in the signaling process, which does the activating of T cells. CD4 and CD8 co-receptors bind to the MHC molecules, and the whole process is able to be seen in the picture shown to the right.
Howdy, my name is Natalie Chavez, and I'll be presenting Chapter 6, Effector Mechanisms of T-Cell Mediated Immunity. What are the types of T-cell mediated immune reactions? T-cell mediated immunity is important to defend against intracellular pathogens. T-cell mediated immunity involves 2 types of reactions of T-cells, which are T-cells that have CD4 plus receptors or CD8 plus receptors. CD4 plus cells, or also known as T helper cells, enhance the functions of other cells through cytokine secretions, and CD8 plus cells directly kill infected or abnormal cells, and are known as T cytotoxic T cells. And here in this image, we can see T helper cells on the top, and T helper cells will recognize or assist in recognizing class MHC2, which is present in antigen-presenting cells, and T-cytotoxic T cells is demonstrated in the bottom, or the CD8 plus T cell. And the CD8 plus T cell will recognize MHC class 1, which is present in all nucleated cells, presenting pathogenic peptides. In Chapter 6, we cover 4 subclasses of CD4 plus T cells and their roles in cell-mediated immunity. There are more than 4 subclasses, but these main subclasses are Th1, Th2, Th17, and TFH. Th1 cells are necessary for macrophage activation by releasing interferon gamma, allowing the macrophage to destroy the intracellular microbe. Th2 cells are involved in the defense against helminths, parasites by recruiting and activating eosinophils and IgE production. Th17 cells recruit neutrophils, whose main function is to fight against extracellular bacteria and fungi. TFH cells, unlike the other previously mentioned ones, they reside in the germinal centers of lymphoid organs, helping B cells undergo affinity maturation and class switching to produce antibodies. Additionally, these cells can be identified based on the cytokines that they release and recognize, and their ability to perform their unique functions. Mechanisms of T cell activating macrophages. Macrophages are important in innate immune cells and require signals from T cells in order to become fully activated. There are 2 types of ways that T cells activate macrophages. In this image, we can see the classical pathway, which is on top, in which macrophages are activated by the release of interferon gamma that is produced by T helper cell 1, which increases the microbicidal activity of the macrophage by activating the production of reactive oxygen species, nitric oxide, and lysosomal enzymes. At the bottom of the image, we can see the alternative pathway, which activates M2 macrophages by releasing interleukin-4 and interleukin-13 secreted from T helper 2 cells. These macrophages are crucial for repair, fibrosis, and anti-inflammatory responses. The responses of macrophages that result in the killing of ingested microbes involved or the employment of many mechanisms to kill these pathogens inside the phagocytes by the secretion of reactive oxygen species, nitric oxide, and lysosomal enzymes. How do CD8 plus CTLs kill cells infected with viruses? The method by which CD8 plus cells kill infected or abnormal cells is via the perforin granzyme pathway. First, a T-cytotoxic naive cell will recognize the MHC class 1 presenting antigen peptides. Co-stimulatory signals such as B7 and CD28 will enhance their activation, releasing interleukin-2, which will self-promote proliferation and effector cells. The CTL will migrate or cytotoxic T cell to targeted cells, and the perforin gram enzyme will be exocytoted. The perforin will form pores on the surface of the targeted cell, and the granzymes will make its way inside, activating capsaicin, degrading the cell from the inside, which would lead to apoptosis. And all of this can be seen in the image, and especially in the middle section of the image, we can see the perforin granzyme being exocytoted and targeted towards the targeted cell, creating perforins on the surface of the targeted cell, which is where the granzyme will then make its way into the cytoplasm, activating capsaicin, degrading the cell from the inside. Thank you, and this is our resource.